Okay, so let's start with the conversation and that the question answer session. Fazila, please go ahead now.
Sir, endoplasmic reticulum is basically made up of two parts, right? Rough and smooth. The rough one is used for protein synthesis, where it is lined with ribosomes. So I have a question that why is DNA or RNA not present in it that is used to create the proteins? Like DNA creates M messenger RNA that further synthesizes proteins.
So your question is that why
RNA or DNA is not present in endoplasmic reticulum.
Okay, so once again, I mean, I love the questions whenever you people, and these are wonderful questions. Fazila Shabash for this question. The question is that if the endoplasmic reticulum is responsible for the synthesis of proteins, then why the DNA is not there? Because the protein is actually synthesized when it is instructed from the DNA, but the DNA is present in the nucleus. So that is the question. The answer is that the DNA is a very sensitive, important, and delicate molecule. So it is required to be placed right within a chamber in a well-protected form. So that well-protected situation can only be provided by the nucleus. Since we know nucleus is a double membranous chamber and the traffic is very controlled, things can go in and out very in a controlled fashion. So that is why the DNA is present within the nucleus. Once the DNA produces the RNA, that RNA goes out of the nucleus and into the cytoplasm for the protein synthesis. Okay, now look at the screen. If you can see this screen, you people can see my screen. Let me full screen this one so it will be easier for you to understand. The job of protein synthesis is for the ribosomes. This structure is responsible for the protein synthesis. If you can see the thread, threads, if you can see these threads are messenger RNA. These messenger RNA came from the nucleus, which carries the information. And these beads, these little circles, they are the ribosomes, this blue thing. So ribosomes are placed on the messenger RNA. Look at that here in this structure. This thread is the messenger RNA. It's very long. It starts from like here, and then go here, and then go. It's very long. And on that thread, there exists the ribosome. The job of the ribosome is to create the protein by connecting the amino acids using the information present on the messenger RNA. So that is how the RNA is utilized to create proteins on messenger RNA. Now, Fazila, this will conclude everything. The rough endoplasmic reticulum contains ribosomes on its surface. So look at those beads, 1 dot, 2 dot, 3 dot, 4, so many dots out there. These dots are actually ribosomes. So what that means? These are the places where protein is synthesized. So actually protein is synthesized by the ribosomes present on the endoplasmic reticulum. And after that, it will be delivered into these chambers. It is delivered into these sacs. It is delivered into these channels from where it can be transported to the other places where the proteins are required. Okay, so since this place is open, sensitive, and open for the degradation, so the very sensitive molecule like DNA is not placed here. But बल्कि आपको एक और बात पता होनी चाहिए. The cytoplasm is a very dangerous place. You know, this is the nucleus. If you can see my cursor, this is the nucleus of the cell. This is the cytoplasm, and anything in between is called a cytoplasm. There exist many enzymes We call them restriction enzymes. नाम भूल जाए खैर है, सिलेबस से बड़ी बात है, याद रखने की जरूरत नहीं है. The restriction enzyme, the job of the restriction enzyme to destroy any DNA which comes into that nucleus, which comes to that cytoplasm. What that means, if any DNA is present here in the cytoplasm, it will be chopped off, it will be cut, it will be destroyed by the restriction enzyme. Now imagine the beauty of that thing. The virus came into our cell, it will enter into the cytoplasm. So the restriction enzymes will destroy that virus. When bacteria came into our cells, it enters into the cytoplasm. The restriction enzymes will destroy that bacteria, the DNA of that bacteria. And the restriction enzyme which we are carrying in our cells are not sensitive for our own DNA. So what that means, for human DNA, the restriction enzymes are blind, but for viral DNA and for bacterial DNA, the restriction enzymes, they are like very much sensitive and they are ready to kill. Okay, hope this answers the question as well as explain the topic. Please, Shabash, let me escape from here. Who is next? Shabash. I can't see actually let me see who is asking the question. Who's next? Now you can see how I'll see you. Anybody wants to ask question about cell structure, function?
Sir, here.
Okay, मीर जलाल शाबाश, आप शुरू कर सकते हैं अपना क्वेश्चन.
सर हम ये देख रहे हैं कि दो टाइप के सेल्स हैं, एक यूकैरियोटिक और प्रीकैरियोटिक, और इन दोनों में बहुत ज्यादा फर्क है, यानी कि एक बहुत प्रिमिटिव है और एक बहुत एडवांस्ड है. तो दरमियान में कोई इंटरमीडिएट स्टेट्स क्यों नहीं है, यानी कि जो आधे प्रोकैरियोटिक से मिलती हो आधे यूकैरियोटिक से मिलती हो, मतलब सिर्फ दो टाइप की हैं, एक बहुत प्रिमिटिव है एक बहुत एडवांस है, दरमियान में कुछ भी नहीं है, तो ये क्यों नहीं है?
ओके मीर, आप जो सोच रहे हैं वो आप एवोल्यूशन के पॉइंट ऑफ व्यू से सोच रहे हैं. जब चीजें इवॉल्व होती हैं तो शुरू में बहुत सिंपलर है, जैसे प्रोकैरियोटिक सिस्टम है, एंड देन आफ्टर दैट इट एंडेड अप लाइक यूकैरियोटिक सिस्टम. So we should have a range of cells with intermediate properties. The answer is we do have the range of the cells with intermediate properties. The range of cells depends on the nature and options of life out there. For example, we need to use one example from the prokaryotic system. Now just imagine, for the prokaryotic system, there exist hell of prokaryotic organisms, large number of prokaryotic, some are very simpler prokaryotic organisms, some are a little bit more complex, and the other ones are multicellular, even made up of many cells. So even within the prokaryotic system, we do have like a different types of prokaryotic system with various degrees of complexities. The same is true for the eukaryotic system, that we have different type of organism with different level of complexities in their cellular structures. That is why you can see, if I tell you that coral reef, did you remember coral reefs which are present in the ocean? The coral reefs, they are living things, but they are made up of very simple cells. The humans are living things, but the humans are made up of very complex type of eukaryotic cells. And there exist many organisms with a different complexity level between the coral reefs and the humans. So there exists a lot of cells with various degrees, and that defines the difference in the organism as well. And even in one cell, even in one organism, like me, like human, like animal, even in one organism, we have different types of cells. Even in one organism, we have different types of cells because they are doing different jobs. The cells of the brain, they should be different from the cells of the heart. The cells of the heart muscles should be different from the cells of the skin because they are doing different jobs. So they have different things, different structures. Okay, Shabash. Who's next? Next question. Shabash.
Sir, I'd like to ask a question, please.
जी जी, आप मुझे पहले बताएंगे, आपका नाम मैं ले नहीं सकता, वो तो नहीं है जिसका नाम मुझे नहीं लेना चाहिए। आपका नाम क्या है?
मुस्ताफा। मुसा।
अमीर, तेरा स्क्रीन नेम कितना मजेदार है। ओके जी अमीर। ये हमें कौन गाने सुना रहा है? Okay, who's next? Who wants to ask?
Sir, I wanted to ask a question.
जी मूसा, आप बात करेंगे?
जी सर।
जी जी।
सर, ये म्यूजिक प्ले होना शुरू हो गया, दोनों कौन?
नहीं, I have been interrupted with somebody. I don't know who is that. I am going to remove that person. आप अपनी बात कर सकते हैं. Okay.
Sir, the first question that I wanted to ask was, what was the purpose of cell wall? क्योंकि आपने बताया था कि the cell membrane, one of its functions is to give structure. So if a cell membrane gives structures, then what is the purpose of the cell wall in plant cells, for example?
Okay. So the question is about the purpose of let me find that thing here. The purpose of cell wall. Since the cell membrane is supposed to present in the outermost region of the animal cell, is present in the outermost region of the animal cell. So if this is enough, that why the plants needs to have an additional wall around them, which is the cell wall. Remember, the plants, they are having a different type of life as compared to the animals, particularly in terms of getting their nutritions. The animals are motile. They can choose to move from one place to another for their food and for other purposes. The plants, unfortunately, they are not. They are supposed to be present at one place for all of their lives. So they need to adopt certain things which can help the plants to preserve important ingredients within their cells. And you know, one of the most important ingredient present in their cell, that is the water. So the cell wall, the additional layer of cell wall in the plants is actually giving them, number one, an additional layer to protect the ingredients. That's the one thing. The other thing is that since the plants are not supposed to move from one place to another, they are more vulnerable for the physical damages. Because they can be touched easily, they can be catched easily, they can be destroyed easily. So in order to have or keep the stronger structure, they need a supportive layer on their cell. The cell wall is there. And not the least, there are many reasons I can list, but just understand that this is an additional protective layer which helps the plant cells to survive as a plant. For example, the trees, the special thing about the trees that they are carrying the bark. They are having a stronger structure which help in spreading their shoots, and helps in spreading their shoots so that they can expand their leaves to receive maximum light from the sun to prepare maximum food. So their structure needs to be very strong, tough, and resilient, so they can take that all that pressure. Whatever the wood you can touch within a tree, these are actually the cellulose. Long time ago, there were cells present in that within those cells, and after that, the cells are dead, and the cellulose become like the wood. So there are many reasons the cell wall is contributing, but it is not like a controlling thing. It is not stopping anything to go into the cell. It is not stopping anything which is coming out from the cells. It is just like I must say, it is just like an extra protective layer which can help the plants to live in a plant way. Okay, so here we go. Next question.
Okay, sir.
शाबाश.
Thank you.
Oh no, कोई thank टीचर को थैंक यू बोलते सवाल का जवाब देने. मुझे खुशी है, मुझे अच्छा लगता है आप सवाल पूछते हैं. Okay, next is, there is a Spider-Man out there. I should mute him, or I should wait to ask his question. I don't know. Okay, next. Fazila, you are asking. Okay, Fazila, आप अपना question पूछ सकते हैं मेरे से. Let me stop sharing my screen and here we go. The next question, if there is, Misha.
Yes.
Okay. You want a question? You want to ask a question?
Yes.
Okay, please.
So you just said the heart और brain के for example cells different होते हैं. तो उनमें basic difference जो है वो function difference है या structural difference है?
Shabash Fazila, it's a very good question, and I used to teach this thing as a professor, like as a major professor of a major subject. That what are the reasons that one type of cell is different from another type of cell? That's a very important question. Why our heart muscle cell is different from the neurons present in the brain, or why one type of cell within our body is different from the another type of cell. So this is the question. Everybody should understand the question, and I always focus on the point that we should understand the question. The answers are fine. So this is the question. The answer is that we are carrying the DNA in each and every cell of our body. That DNA is perfectly identical in all cells. If the DNA present in all the cells are 100% similar, then how it is possible that we are carrying that much different type of cells using the same DNA? Another thing, if one cell is different from another cell, like we can see the skin cells are different from the eyes cells are different like from our blood. The blood cell is different from our other cells present in the body. If two cells are different from each other, that means these two cells have different structures. They have different proteins. Now the critical question is that how it is possible that two cells with the same DNA is showing different structures. If you are getting this question, you are fine. If you are getting this question, so the question is that I am human, all of my cells are carrying the same genetical material. All of my cells are carrying the same genetical material. How it is possible that one of my cell is different from another? Because I can see the eye cell is different from the tongue cell is different from the nose is different from the skin. How it is possible? To understand that thing, they have the same DNA. They have 100% identical DNA. To understand that thing, you need to understand the concept that imagine a piano keys. The piano keys has a certain number of keys. I mean, I don't remember. I don't know, the 24, 23, 22, whatever. The piano keys has a certain number of keys, and you can create different tunes using the same keyboard of the piano by hitting and by striking the same keys in a different way. अलग रहे समझ आ जाए. Are you getting my point? Using the same piano keys, by hitting and striking it in a different way, you can create different tunes. So our cells with the same DNA, using the same DNA in a different combination to produce different number of proteins, create different structures and different cells. So the beauty lives in our complexity arises because our cells choose to utilize the same DNA in a different way. Okay, what's next? This all ये बहुत important सवाल है. Okay, they should go to the YouTube to answer the people at 100 level. Okay, शाबाश. अगला सवाल. Next. जी रिम से शाबाश आ जाओ. Why I can't see your microphone turn on?
सर मैंने अभी पूछा था आपको.
अच्छा मिशा ने पूछा था. Sorry.
Yes.
Okay, who's next? No? Oh, anybody wants to ask the question? Okay, that's it. So now you need to know, what you need to do is that you people need to download the video lecture as well as the slides, and then you are supposed to prepare those things and wait for me. So I should give you some form of assessment so that I can analyze and I can give you a certain grade based on your performance. And I'm not doing it in this week, since I want you to climatize yourself for this new form of education, and hopefully I will be giving you certain type of assignment during the weekend or on next Monday. Okay, so that we will start. I will start grading your performances as well in the meanwhile. जी मीर आप कुछ कहना चाहते हैं?
Sir, I have a question about ribosomes. Since ribosomes are responsible for protein synthesis, it's rather difficult to imagine that what happens when cell is in the process of becoming. Which structure is responsible for creating proteins when cell is not complete, when there are no ribosomes?
Shabash. It's a very good question, and the answer is that, for example, if the cell is planning for the cell division, if a cell is planning, a cell is just born and not having all the things. So when the cell is not there, who is responsible for the process of protein synthesis? So the answer is that when a cell become adult and decide to go for the cell division, it needs to divide and it needs to produce new cells, daughter cells.
यार पापा से पैसे पूरे मैंने अगले मंथ लेने, मैंने बालों पे क्या.
In the meanwhile, oh my God. In the meanwhile, the cells do not exist as a cell. When they plan to go for the cell division, and when they produce the daughter cells, in the meanwhile, they cease to exist as a cell because their cell membrane is gone, their other membranous bounded structures are gone. So in that time, they cannot act as a cell. So यहाँ पर मीर का question है, this is the time, so how the cell will pass or live in that state when it is no longer acting as a cell. So the answer is that when the cells become adult and they choose to go for the cell division, the cells prepare all the necessary protein for that purpose. And after that, there exists no protein synthesis, and cell is dependent on the proteins it has created earlier, and then use that protein to execute the function which I was talking about, the cell division and other functions. That's very important, and it is even much more important if you want to think in a very difficult phenomena, reproduction. Remember, the sperms are produced somewhere else, the eggs are produced somewhere else. The sperm needs to find the egg and then fertilize it, and then go for what? And go for the cell divisions after that and develop into a baby. So for that particular time, there exists no ribosome, and the cell is dependent on all the protein which is supplied by the initial cells. Or you can imagine another important example like the egg. The egg contains one cell, and after that the zygote one, when it was there, it was not like whatever the zygote needs for its development is there within that zygote, or it is provided by the system. So all the proteins which are required for the development of the zygote is provided in the egg. So egg can use them and then develop into a bird. Okay, shabash. Next. So good, these are good questions. Okay, you received the video lecture of chapter number 4 as well. Okay, I will be recording, I will be recording the lecture number 5 for you people to save time and to give you some extra credit hours. Actually, we have like we are using more credit hours than our natural or normal credit hours we supposed to consume in a week, because I am recording the lectures as well and I am talking to you as well. So there are like 2 lectures for every class we are having. Okay, so don't worry, chill about these things. Okay, any question?
Yes, sir, I have a question.
Ji shabash, ji puchiye.
Sir, the job of lysosomes is to protect the cells and digest the food material. So
Ji.
can lysosomes destroy the virus that comes into the cell through exocytosis?
Okay, virus is too small. Virus is too small for the lysosomes to deal. I mean, virus is like is too small for that thing. Not possible for a lysosome to capture or to catch the cell. The picture was not there. I have recorded a picture of my drawing at that time for you people to understand how lysosome actually created and how lysosome capture the things. There are 2 purposes for the lysosomes. Number 1, if a food touches the cell membrane, it invaginates, create a food vacuole, and then after that food is required to be consumed. The first purpose. The second purpose of the lysosome is that if there is something which is not healthy, like the bacteria or a bigger component, a bigger bad thing, so the cells can consume it. What happens, for example, if the cells engulf a bacteria, what will happen after that? Here we go. So yes. The question was about the lysosome. Fazil, am I right? Fazil.
Yes, sir.
Fazil, आप repeat कर सकते हैं अपना question. Okay, confusion.
Sir, the question was that can lysosome destroy the virus through exocytosis?
The simple answer is that the virus is too small for the lysosomes to deal. Okay, it's too small. उसको पता भी लगेगा कहाँ से आया कहाँ चला गया, तो वो उसके बस की बात नहीं है. But for the bacteria and the other, the answer is yes. Okay. Okay, let's move on to the next question. If anybody wants to ask a question, do we have another question? Okay. So let me tell you about the next class, and then you are done with this session. And I will be talking about what I will be creating for you. Okay, as the next lecture. और हम क्या पढ़ रहे हैं? As I told you, that one of the student is missing. I think it's not here, but I think I have sent the meeting ID to our class group, and then these people can join me. Okay, here we go. I will be talking about, I'm going to share my screen for you people, and I will be talking about the chapter number 5. Okay, so this is the chapter number 5 we are talking. The name of the chapter is the cell in action. So it is another important lecture chapter. I will be talking. You are not supposed to study today from me from here right now. Okay, right now you need just to know about the lecture and the chapter, and then I will be sending a video for you people, and you people will going to browse the slides as well as listen to the video and try to understand that things. And if you have any problem, we will be addressing that problem in our next session. Okay, so that's what we need to do. Okay, here we go. Okay, here we go. So what about the what's there in this chapter? I will be telling you in like 2 or 3 minutes, and then you should wait for me to create a video lecture for you for this for chapter number 5. The chapter number 5 will be about the cells when the cells are doing certain jobs, when the cells are doing certain functions. For example, how the cells talk to their environment, how the cells talk to their external conditions, how the cells exchange the things between themselves and with the environment, how the cells absorb oxygen, how the cells like release waste products out of them. So how the cells communicate with the environment, we will be studying that thing. What are the principles, what are the laws which governs these exchange of material between the cells and between the environment? We will be studying those things. Diffusion is a very simple principle, and then osmosis is very, very, very important. इसके बारे में क्वेश्चन जरूर आएगा, okay? And then जैसे मैं आपसे अक्सर बताया करता हूँ कि whenever somebody create an assessment about this chapter, he or she will be stopping at osmosis. So when I will be talking about the osmosis, you need to be very careful and try to help yourself to get these things like completely. Like the other means of transportations, passive and active transportation, they are very easy. I will be talking on these things. And then endocytosis, ये हमने पढ़ लिया, बस इत्तेफाक की बात है. ये वो स्लाइड है जो मैं ये वो ड्राइंग है जो मैंने ड्रॉ की थी आपके लिए लास्ट लेक्चर में. What is endocytosis? What is exocytosis? And then how cells get their energy, from where the cells get their energy, and then how plants create their foods, and how the rest of the organisms consume that food. What is respiration? If you can remember, it's a mode of producing energy. It's a way of consuming food. How we will go into the detail of that mechanism, and we will study how mitochondria actually produce energy. Okay, and then what is fermentation? What is cell cycle? Cell cycle is about the life of the cell. If you try, you can imagine the life of the cell. When cell is created, it tries to grow itself, then it will become adult, and after that it go for the cell division once again. This is what we call as the cell cycle. And we will be studying about the comparison between prokaryotic and eukaryotic system, and I believe you people understand that thing. And then the most wonderful thing is about the cell division, the prokaryotic cell division and the eukaryotic version of cell division. Actually, we are supposed to study about these things in lab as well, but since we are not going to have a lab in this way, so I need to create a certain experiment for you, which I will be considering as your lab exam. Okay, वो मैं बाद में बताऊंगा, लेकिन अभी नहीं. इस वीक में सिर्फ हमें ऑनलाइन क्लासेस को और उससे जुड़ी हुई कॉम्प्लिकेशंस को समझना था, जो कि भी हम समझ रहे हैं. Okay, and then we will be talking about the DNA, how it is divided, what are chromosomes, and cell cycle in prokaryotes, and cell cycle in eukaryotes, and then the most famous thing, mitosis. Okay, I might not be including mitosis in our content. This is a good news for you because we will going to reduce the content. So some of the important things might be deleted as well. So maybe mitosis will not be there. So expect me to talk on these slides and to create a video lecture for you. And I will be giving you all that material along with the slides for your study. And you can take your time like 2 or 3 days or on Monday to create your questions about these slides, and then you can ask your questions to me. Okay, and for your semester as well. Okay, किसी के पास कोई और question है? We are almost done with the weekend. I am stopping the recording for you, but before that, Mir wants to ask a question. Ji, Mir.
Sir, how many chapters are we supposed to cover?
Actually, it depends on our speed and nature. We are not stick with anything. Frankly, we are dependent on the speed of the 7 and 8 sections we are having. ठीक है, तो मजे की बात है कि अगर फर्ज करते हैं कि सारे लोग मिलके सिर्फ 2 चैप्टर ही पढ़ते हैं, और तो हम 2 चैप्टर से ज्यादा नहीं पढ़ सकते. Are you getting my point? तो हम दुआ करेंगे कि कोई भी ना पढ़े तो हम भी ना पढ़े. It is high chance, but I will be pushing, because whatever the chapter I am recording, we will be sharing those chapters as well with the other students from the other students can also access those things. So university has a wonderful way. If somebody like my students can learn from other teachers, and other teachers, the students from the other classes can learn from us. But it will be like whatever everybody else is learning. Like everybody else is like learning like 5 chapters. We will go ahead with the 5 chapters only. If we all like learn like 7 and 8, it depends on the naturally कि हम पढ़ते हैं कितना है. But that is like, as an instructor, it is my job to see that from this chapter, these are the less important things. So I should remove that content. I should focus on these 1-2 topics better than other, because we don't know, we are not used to of this method enough. After 2 or 3 cycles, we might be very efficient. We know, okay, these are the things we need to talk. That is how we need to teach. The online lecturing is the best way, or the recording options are the best way. So these are all the things which we need to evolve in the meanwhile. So the content depends on how the other classes are going with us as well. So together, whatever the content we will be creating, हम उसके मुताबिक ही वही आपका syllabus होगा. ठीक है? I will make sure कि आपके पास कम ना हो. Okay, this is my job. कि whatever the rest of the students are studying, you should be able to learn at least that much or more than that much. ठीक है?
सर आपने गूगल जो बनाई हुई है वो क्लासरूम, उसमें सर आपने एक लेक्चर अपलोड किया है तकरीबन 30 मिनट का, लेकिन जो मूडल है सर उसमें 3-4 वीडियोस अपलोड हुई हैं. तो ये समझ नहीं थी आ रही कि कौन से मतलब ज्यादा बेहतर है, कौन से लेना चाहेंगे?
मेरे ख्याल से दोनों में एक जितना ही है. आप गौर से जाके जब दोबारा से ब्राउज करेंगे, गूगल क्लासरूम में जो प्लेस करने के ऑप्शंस हैं वो मूडल से अलग हैं. जब आप गूगल क्लास में क्या आप देख पा रहे हैं? मेरी स्क्रीन देख सकते हैं ना आप इस वक्त? क्या आप मेरी स्क्रीन देख सकते हैं?
यस सर, देख सकते हैं.
ओके, मैं आपको दिखा रहा हूँ. ठीक है, क्लासरूम में जाके वो जो आप 2 चीजें देख पा रहे हैं, the two things which you, for example, I have 2 classes. Okay, this is a BIOL100 class. Okay, I'm here as a teacher. So you मेरा जो देखने का अंदाजा है वो आपसे अलग होएगा. ठीक है, इस वीडियो. But you should be able to see the classroom in this way. There should be 2 topics over there. The one is the online lecture number 1, that is about the pandemic. The other is online lecture number 2, that is about the chapter number 4. Now, if I click on these things, on this topic, for example, online lecture number 2, it will take me to the components जो इसके अंदर मौजूद हैं. इसमें ये वेब लिंक्स मौजूद हैं. अगर मैं इस टेक्स्ट फाइल को खोलूंगा तो इसमें वो सारे इंटरनेट लिंक्स हैं जो मैंने इस्तेमाल किए हैं. कोई यूट्यूब शॉर्टकट है, यानी कि ये एक शॉर्टकट है जिसकी मदद से आपको सारे लिंक्स मिल जाएंगे बजाय उस वक्त डाउनलोड करने के. यानी कि अगर आप चाहें तो अपने मोबाइल के ऊपर भी डाउनलोड करके कहीं ऐसी जगह पे जा सकते हैं जहां पर इंटरनेट मौजूद है, तो इन लिंक्स को इस्तेमाल करके आप डाउनलोड कर सकते हैं. ठीक है, उस पर आप देखेंगे ये 4 वीडियोस हैं इसके नीचे. तो ये सिर्फ ब्राउज करना और ऑर्गेनाइज करने का डिफरेंस है. ये मुश्किल है, लेकिन आई हैव प्लेसिंग द थिंग्स इन इक्वल वे इन बोथ द प्लेस. सो यू कैन सी द 4 वीडियोस आर हियर. अगर ये पहले वाले पेज पे विजिबल नहीं आ रही थी, लेकिन वो इसके नीचे हैं. तो जब भी आप एक टॉपिक पे क्लिक करेंगे, अगर मैं ये 2 टॉपिक्स हैं, तो जब इस टॉपिक पे क्लिक करेंगे यू एडेड अप लाइक विद ऑल द अदर थिंग्स प्रेजेंट इन दैट टॉपिक. तो मैंने इनको इस तरह टॉपिक वाइज अरेंज किया. जैसे अब हम एक्सपेक्ट करेंगे इसमें चैप्टर नंबर ऑनलाइन लेक्चर नंबर 3 ऐड हो जाएगा, तो उसमें उससे एसोसिएटेड पिक्चर्स. अभी जो हमने कन्वर्सेशन की है इसके जो इंपॉर्टेंट कंपोनेंट्स हैं या इसमें जो इंपॉर्टेंट क्वेश्चन एंड आंसर्स हैं, मैं उसको एडिट करूंगा. यार ये सबसे मुश्किल काम है जो मैं करता हूँ. मैं इन कंपोनेंट्स को एडिट करूंगा और एडिट करके यानी कि उनको क्लिप्स में से निकाल के यहाँ पर आपके लिए प्लेस कर दूंगा, ताकि जो दूसरे स्टूडेंट्स हैं अगर उनके पास भी इसी तरह का क्वेश्चन है तो वो एड्रेस हो जाए. वो तो दैट इज हाउ एवरीथिंग इज देयर इन द सेम. ठीक है, मुझे समझ आ गया? ओके
सर वो काइंडली अमीक को जरा व्हाट्सएप ग्रुप में ऐड कर दीजिएगा. उसका नंबर मैंने पहले भी भेजा था तो वो ऐड नहीं हुआ. ठीक है, मैंने अपनी तरफ से अमीक को ऐड किया हुआ है. मुझे लगता है मैंने किया हुआ है. मैं दोबारा कर देता हूँ. ठीक है सर, थैंक यू. मैं उसको दोबारा, वैसे मैंने लगता है मैंने आपको किया था, उसको मैं दोबारा कर देता हूँ. थैंक यू. और जी
अब जो ग्रेडिंग क्राइटेरिया होगा वो कैसे होगा? मतलब क्लास पार्टिसिपेशन, अटेंडेंस वगैरह की, अब वो वेटेज तो नहीं होगी. अब उसकी क्या वेटेज किस हिसाब से?
शाबाश, थैंक यू एहतशाम। इस सवाल का आंसर मैंने लास्ट टाइम में किया था और मैंने अपने एक लेक्चर भी आपको थोड़ा सा दिया था, ओपनिंग लेक्चर था कि ऑनलाइन टीचिंग में होता कैसे है। तो ये बिल्कुल वैसे नहीं होगा जैसे आपने पहले देखा हुआ है। जो आपको कोर्स आउटलाइन मिली थी उसमें जो जो डिवीजन थी ग्रेड्स की, ये बिल्कुल वैसे नहीं है। ऑफ कोर्स इसमें जो क्लास पार्टिसिपेशन या कंट्रीब्यूशन है वो बहुत वैल्यू रखती है, क्योंकि अब ये ऑनलाइन टीचिंग है। आप कितना पार्टिसिपेट करते हैं, आप उसमें कितना कंट्रीब्यूट करते हैं, आप कितने क्वेश्चंस बना के लेके आते हैं, आपके क्वेश्चंस की स्ट्रेंथ क्या है, उनकी क्वालिटी कितनी है, और उसके बाद असेसमेंट्स हैं जो हम आगे डिजाइन करेंगे और मैं आपके लिए आगे लूंगा। तो अल्टीमेटली ये बिल्कुल ऐसा नहीं होगा, इसमें मिड टर्म्स नहीं होंगे, इसमें फाइनल टर्म्स नहीं होगा। एवरीथिंग डिपेंड्स ऑन द स्मॉल टाइप ऑफ असाइनमेंट्स व्हिच आई विल बी गिविंग टू यू। तो हर एक चैप्टर से जुड़ी हुई कोई ना कोई असाइनमेंट जो है वो आपको मिलेगी, जो जैसे जो ऑनलाइन लेक्चर 1 को आपको डिफेंड करना पड़ेगा, ऑनलाइन लेक्चर नंबर 2 के बारे में कुछ सवालात को आपको डिफेंड करना पड़ेगा। तो हर एक लेक्चर से जुड़ी छोटी छोटी असाइनमेंट्स होती है, और मे बी एट द एंड। जी जी जी
प्लीज सर आप।
हाँ, तो छोटी छोटी असाइनमेंट्स इंडिविजुअल लेक्चर्स के बारे में होंगी, और वो तमाम असाइनमेंट्स जो मैं आपको दूंगा, इट शुड नॉट बी द सेम असाइनमेंट्स व्हिच यू यूज्ड टू हैव अर्लियर। मैं आपसे ये नहीं कहूंगा कि जाओ जाके 15 सफे जो है वो टाइप करके मुझे सेंड कर दो, या जाओ जाके इसके बारे में ये, क्योंकि यू नो गूगल कॉपी पेस्ट इंटरनेट ऑल दिस थिंग्स आई नो। आई नीड टू क्रिएट योर असाइनमेंट्स और क्वेश्चंस इन सच ए वे ताकि मैं आपकी परफॉर्मेंस को और आपके कॉन्ट्रीब्यूशंस को जज कर सकूं। मेरे लिए जरूरी ये है पता लगाना कि whoever is trying to understand, असल में किसी भी कोर्स का जो पर्पस होता है वो नॉलेज को हासिल करना होता है कि क्या आप वो चीजें हासिल कर पा रहे हैं। मुझे वो चीज चेक करनी है कि वेदर यू आर एक्चुअली गेटिंग द थिंग्स व्हिच आई वांट यू टू लर्न इन द मीनवाइल, और यू आर पुटिंग योर एफर्ट टू अंडरस्टैंड दोस थिंग्स और नॉट। सो इस चीज को मैंने मेक श्योर करना है, तो मैं इसके ऊपर ऐसी असाइनमेंट्स क्रिएट करूंगा। और ऑफ कोर्स आप एक्सपेक्ट कर सकते हैं कि बिफोर द फाइनल situation I might be asking like a bigger assignment, I might be asking about a bigger assessment method like for you. मैंने पहले भी मिसाल दी थी, दोबारा देता हूँ कि फॉर एग्जांपल if I have delivered a lecture or I have given you a video lecture or I have given you a YouTube link, मैं आपको YouTube के ऊपर एक लिंक जो है ना वो फॉरवर्ड करता हूँ, and then I will be asking you people to please come forward here and start answering a question. मैं आपसे एक सवाल करता हूँ, फॉर एग्जाम्पल ये सब मैं आपको, what is the most interesting thing you find in that documentary or you find in that YouTube link? Now it depends on your understanding, it depends on the way of looking at certain material. You observe, you study the video, and then you start answering me about your impression. From your impression I should be able to understand how much you actually know about the material present in your classroom. So that is how I can't ask you the questions like this, which of the following is not an example of cell organelle, because I will be giving you the MCQs. The people are out there, the internet is out there, these methods will not work. So we need to design certain innovative method to judge your performance in an honest way, ताकि आप भी फील करें ना कि मुझे अच्छे तरीके से जज किया गया है, मुझे प्रॉपर सिस्टम प्रोवाइड किया गया है। तो जो भी असेसमेंट मेथड मैं चूज करूंगा, मैं पहले आप लोगों को एक्सप्लेन करूंगा। फर्स्ट मैं आप लोगों को एक्सप्लेन करूंगा कि ये वाला असेसमेंट मेथड है, इसमें क्वेश्चन इस तरह से पूछे जाएंगे, आपको आंसर इस तरह से करने हैं, ताकि आपको बिल्कुल क्लैरिटी हो उसको आंसर करने की। ज्यादा जरूरी बात क्या है कि आप तक नॉलेज पूरी तरह से पहुंच जाए, आप तक जो भी मैं टॉपिक शेयर कर रहा हूँ वो किसी ना किसी तरीके तक आप तक पहुंच जाए, चाहे वो क्वेश्चन आंसर सेशंस हैं, चाहे वो मेरा वीडियो लेक्चर है, इट शुड रीच टू यू, इट शुड फाइंड्स यू। और अगली बात क्या है कि हम अब आपने उसको प्रिपेयर करना है और फिर मैंने उसको असेस करना है, तो मेथड्स विल बी डिफरेंट। शाबाश। और किसी के पास कोई कंफ्यूजन है ना, बगैर किसी वजह के ट्रबल के आप मुझसे पूछ सकते हैं। दुआ आपको अगर कोई बात पूछनी है, फहीम आपको पूछनी है, राबिया इज हियर, मूसा इज हियर, मीर, कोई सोलोमन, किसी किस्म का कोई कंफ्यूजन है, यू शुड आस्क, ठीक है? इट हेल्प्स योरसेल्फ एंड योर फ्रेंड्स एज वेल।
यस सर।
जी जी
Sir, already cleared. You have cleared everything. I just watched the last video also, so no more queries about anything.
थैंक यू वेरी मच। लेकिन एज ए राबिया रिमेंबर व्हेन यू विल बी अ टीचर इन द फ्यूचर, अगर आपने चूज किया, या आपकी पर्सनल चॉइस है, रिमेंबर द मोस्ट इंपॉर्टेंट थिंग अबाउट द टीचर, you need to keep a very high level of patience in order to act as a teacher. Okay, अगर आपने चूज किया, आप मेरे से प्रॉमिस करेंगे कि आप इतना हाई लेवल ऑफ पेशेंस मेंटेन करेंगे. ओके,
Yes, sir. Yes,
क्योंकि
sir.
आपको एक ही बात इतनी दफा बतानी पड़ती है, इतनी दफा बतानी पड़ती है, that is actually test the level of your patience. Although I have like created
Yes.
3 platforms, but I am absolutely fine with that. मुझे ये बुरा नहीं लगता है. I love that thing. मुझे पता है कि स्टूडेंट्स कभी-कभी सिर्फ इसलिए सवाल नहीं पूछता है कि अगर मैंने सर से पूछा तो वो कहेगा मैंने 4 दफा तो बताया है, 5 दफा तो बताया है, और फिर आप मुझसे पूछ. और आपको मैंने शुरू में कहा था ना क्लास में कि टीचर्स का लेवल ऑफ पेशेंस पहली क्लास में पता नहीं लगता. पहली क्लास में वी आर ऑल मेरे जो जितनी दफा मर्जी पूछो मैं हूँ.
Yes.
And after that,
Yes, sir.
when you ask the same question in the second class, the most मैंने
Yes,
पिछली क्लास में तो बताया था, तुम सोए हुए थे उस वक्त.
Yes, sir.
So
Yes, sir.
the patience is the most important ingredient for every teacher, and that is fine. I mean, I'm fine with that thing. Just try to be like
Yes,
this
sir.
in the future if you
Yes, sir.
choose to. Anybody else? Okay, so we are ready to conclude. अगर आपने कोई बात पूछनी है मेरे से, अगर आपको लगता है कि आपको मुझसे कोई और questions पूछने हैं, you can be online with me. You can ask for the Zoom session as well to me. You can come on the WhatsApp. You can catch me on the WhatsApp. मतलब मुझे लगता है मैं सारा दिन सुबह दोपहर शाम ऑनलाइन रहता हूँ, ठीक है ना? कोई मसला नहीं है. You can ask me any question about the things. You can catch me here on the Zoom कि सर मुझे ये बात पूछनी है, ये स्लाइड के बारे में क्वेश्चन करना है. So I will try my best to make myself available as much as possible. Okay. बाकी ह्यूमन लिमिटेशंस हैं, कभी-कभी हमसे नहीं होता, कभी-कभी हम कैच नहीं कर सकते, लेकिन आई विल ट्राई माय बेस्ट टू बी विद यू. पर्पस ये है, एजेंडा ये है कि आप नॉलेज हासिल करें और ताकि आप अच्छे से ग्रेड क्लेम कर सकें. ओके, so all set, it's time to conclude the class. I am stopping my screen share with you people, and I am saying goodbye to each and every